Good afternoon, everybody, and welcome to today's webinar. Today we're going to be talking about accurate variant classification and clinical impact of variability. This is a non-promotional webinar organised and funded by AstraZeneca and MSD. Next slide, please. As per normal in these webinars, it falls to me to do the housekeeping and the introduction. So we have automatically muted your microphone. Please do refrain from utilising your microphone at any stage during the webinar. We're recording this webinar and we'll make it available to you on the EMQN and GenQA YouTube channels. Usually takes about 2 weeks before that's available, but we will notify all attendees when that's available. But most importantly, this is a interactive session and we have a Q&A session at the end. Please use the Q&A session to really ask the questions you want to know from the experts in today's webinar. Next slide, please.
Yeah.
Your feedback is also important to us. It's not just obviously us feeding back to you, but it's you feeding back to us about whether you think these webinars are productive and useful for your training and education. We try to make them as high quality as possible. So after this webinar, you will receive an email sent out by the organisers with a link to a very short feedback survey. It usually takes no more than 2 minutes to complete it. I would strongly encourage you to do so, please, afterwards. Feedback your responses to us and help us improve our ongoing webinar program. Next slide, please. Okay, without further ado, then I'm going to hand over to my co-chair, Professor Sandi from GenQA. Sandi, over to you for the introduction.
Thank you, Simon, very much indeed. And I'd just like to join Simon and welcome you to this webinar. This is webinar 3 of our series of 4 that will be delivered during 2025. I'm joined, as you have just met, Dr. Simon Patton, who's the from EMQN CIC, the CEO. And we also are joined by 2 experts today in the field of variant classification, particularly in HRR genes. We are joined from the Minnesota, Dr. Fergus Couch, so welcome Fergus, and also Professor Thomas van Overeem Hansen, who is from Denmark. So next slide, please. These are the disclaimers and disclosures. Next slide, please. So, as you're aware, as with the previous BRCA runs and the HRR gene variant classification assessments, this is delivered using the GenQA Genie platform, which is the Genomic Online Individual Education platform. The difference this run, we have listened to the feedback we have received from you, and we've made it much simpler to be able to register for these participants for these EQAs. So basically what we have done is made sure that you do not need to repeat the registration process for each run. So we're now on run 15. So those of you who have been with us right from the start will remember the long periods of time where you have to go through all the enrollment process. So we've now reduced that, and you just register once, so you get information for each run as it goes live. You can participate even as an individual, or you can do that through your laboratory, and that's existing or new staff. And to make it easy for you, there are now clear instructions and also a how-to video which you can access once you log into your Genie account, and that's through the YouTube channel. Next slide, please. So in a nutshell, just a quick snapshot for purchasing. If you're an individual, you log into your account, you go to the purchase interface, and you select your Genie BRCA Runs product, and you pop it in your basket, and you complete the checkout. So these EQAs are funded through AstraZeneca and MSD through an educational grant, so no payment is required. And the assessment will open to everyone who is registered on Wednesday, the 1st of October. If you're purchasing through your laboratory account, again, you log on with your lab manager doing this on behalf of you, select your manage and manage staff, and then edit icon. You can then edit with the relevant staff member to give them access. Again, you can go through the select and process payments and click the update button. And again, no funds will be required or taken from your account. So hopefully this will be much more user-friendly for you. Next slide, please. So this is the agenda that we have arranged for today. We've got the functional evaluation and clinical classification of BRCA2 variants, and a review also of the learnings from Run 14, which was delivered earlier in 2025. So it now gives me great pleasure to hand over to Fergus. Next slide, please, to give us the functional evaluation and clinical classification of BRCA2. Fergus, now over to you. Thank you.
Okay, thank you. So thanks to the organizers for inviting me to do this. Looking forward to it. Good group of people on here. So I'm going to talk, as Sandi was just saying, I'm going to talk about the functional evaluation and clinical classification of BRCA2 variants. And when I say BRCA2, I'm not doing the whole gene. I'm doing the key functional domain, which is the DNA binding domain, which is down towards the C terminus. This is really where all the pathogenic missense variants have been identified. So this is why we focused on that hotspot region. Sorry, went too far. Here are my financial disclosures. Now, a little bit of background on the BRCA2 gene. As was mentioned already, it has a critical role in homologous recombination. Inactivating mutations in BRCA2, mainly protein truncating variants, result in approximately 69% lifetime risk of breast, ovarian, prostate, and pancreatic, and some emerging evidence of some other cancers as well. But perhaps as many as 1% of breast cancer patients in the general population are carrying inactivating mutations in this gene. Here's just a diagram of the gene at the bottom, and you can see in the red box there where I'm talking about the DNA binding domain. This is, again, the key element where all the known pathogenic missense variants are located, and the reason for our focus. Okay, so really what we're talking about here is classification of variants, so variants of uncertain significance. If you go into ClinVar today, which many people will use as their the database of guidance here, there are over 5,000 VUS. Many of these are missenses, and they remain unresolved. And again, what is the problem for clinical management? It's basically that they don't benefit from the same interventions and management strategies that somebody with a known pathogenic mutation can do. And so this is interventional prophylactic surgeries to reduce risk, MRI screening, at least in the United States, MRI screening, in addition to mammography, cancer risk assessment, further cancer risk assessment for the patient and the family members, and then potentially treatment options such as PARP inhibitors in the context of a pathogenic variant. So they've lost all that ability. So we're faced with 5,000 variants. We're faced with 1,500 of them located in the DNA binding domain. And again, because that hotspot region of the protein and of the gene is thought to harbor quite a large number of pathogenic missenses, we decided to focus in on this in some detail. So this is just a schematic of how we did this. It's a CRISPR-based technology where normally with CRISPR, we delete away pieces of the gene. In this case, we're actually putting in point mutations, putting in single nucleotide changes into the endogenous gene within cells. We use hap1 cells, which are haploid. They have one copy of the genome. This makes it convenient for us because we only have to insert the mutation once into the cell. We don't have to deal with the diploid state. Each experiment is an individual exon. We target the exon. We do each experiment in triplicate at a minimum. Essentially what we do is in plasmids, shown here on the left, we make every possible single nucleotide change in that exon. So we end up with hundreds of different plasmids altogether. We take that whole pool and insert it into the hap1 cells. It targets into the endogenous locus and replaces the wild type version with whatever variant is being inserted. And then we let the cells grow for a little while. And at day 5, which is in essence really day 0 for us, and at day 14, we harvest the cells. And it's the same culture, so they're directly clonally related to one another. We then take the genomic DNA from those 2 points, sequence it in great depth, hundreds of millions of reads, and then literally ratio. So for each variant, how many times did I see it at day 5? How many times did I see it at day 14? We make a ratio. We do the log 2 of that ratio to get a distribution, which is this peak here. And then as part of our process, we go through with the ACMG AMP classification, which you may use other terms, but the Enigma-based classification, the CanVig-based classification, whichever models are out there, we use the functional data as part of that process to classify variants. So that's an overall schematic. You get the idea that you have to do each of the regions. Some of the exons were a little bit larger, so we had to split them in half and do 2 separate experiments. But essentially, as we go across this whole thing, we do many thousands of variants and leave no stone unturned. So this is what the results were like when we compile all the exons together. We do take the data and normalize it across all the exons so they all could be combined together, even though they're separate experiments. And again, the triplicate data is accounted for in here. And essentially what you're looking at is just the depth of the variance on the vertical axis versus on the functional score, which is the log2 of that day 14, day 5 ratio is really what you're just looking at there. And you can see there's a nice 2-component distribution there. The nonsense variants, so the protein truncating mutations, are in that orange color. And the light blue or teal color on the right are the synonymous variants, the variants that don't change the amino acid. And you can see there's a very nice separation of those two pools. So they are essentially our controls here. They separate nicely. And then everything else, predominantly the green color, which are missense variants, are separated out into what looks like 2 peaks with a little group in the middle. So the experiment worked very well. We got very nice separation. And I will just describe some of the findings here. Before I do that, however, I have to convince you that this experiment, even though it looks nice on a picture, it's actually meaningful. So we approach this with 4 different series of standards. They do overlap a little bit, but these are known pathogenic and known benign missense variants, and also known standards from other functional assays. And we evaluated them across the results from this CRISPR study. And you can see on the far right-hand side, you can see the sensitivity and specificity. So really very good, around 95%. The ClinVar standard's 99% sensitivity and specificity. However, it's not perfect, right? There is a little bit of error in there, probably 5%, which you would expect from a biological system like that. It actually, to me, it's remarkable to me that we can get such high quality out of such detailed experiments. But it did work at this level across these 4 sets of standards. And then here's a busy box, but really what I'm calling your attention to is the missenses across the bottom. So as part of the data, you have the functional data that I just showed you. We then convert that into a likelihood ratio, which also can be converted into a probability of pathogenicity. So a likelihood ratio of pathogenicity, a probability of pathogenicity, and that breaks down then into 7 categories across the top here. And these are the categories that are used in the Bayesian likelihood framework that people have applied to the ACMG guidelines. So many of you will be familiar with the pathogenic strong and the benign strongs on the left and right here, and all the categories in between. So what we find here is that over 10% of the missense variants in this region turned out to be in the P strong. So very high probability of being pathogenic in this setting. And then almost 80% way down there in the benign setting, and then a few scattered in between. And the experiment did actually yield information on canonical splice sites as well. So we think of the +1, +2, and -1, -2 positions around the edges of exons. We generally think of those as being pathogenic when they're disrupted. But as you can see here, there's a smattering of them that fall into the middle categories. But for the most part, they do behave. And strangely, then there were 10, so 6.5% of the canonical splice sites that look like they don't do anything at all, that they were actually benign. So we always have to keep that in mind, that there are some that don't work, that don't actually influence splicing. Here we take the types of variants and we break them out and we say, okay, how do they break out by these 7 probabilities or these 7 likelihood ratios? And so if we look at nonsense or protein truncating mutations, we expect them to be highly likely to be pathogenic, and sure enough, almost 100% of them are in the P strong category. That makes perfect sense. The synonymous at the other end, we expect them to be predominantly benign, and sure enough, they're all blue, and that makes sense. And then the missense are a grouping there along the lines of what I just told you before, about 10% looked like they were in the P strong category. So everything looked really clean. We also then decided to compare this against other functional assays, so much smaller functional assays of maybe 100 to 200 variants that other groups had done. In fact, the one on the left is our own homologous recombination functional assay compared to this what we call MAVE, this CRISPR study. And you can see here the p-values are very strong across all of this. This really looks very good. These high quality assays, there's many assays that have been done, maybe over 30 different functional assays have been described for BRCA2, but these would probably be the 4 best. And there's really high quality, at least in the sensitivity for the pathogenic variants. There's really high concordance between the various experiments. So that was nice to see as well. We then take the functional data and use it as part of the PS3 BS3 rule within the ACMG ClinGen classification model. So we just put it in there as another source of evidence. We give it +4 points. For those of you familiar with the point system, you have to get to 6 points to be likely pathogenic, and we give 4 to the functional. Now, the functional assay, the way I just showed you with the sensitivity and specificity, it actually would qualify as very strong evidence and would normally get much higher points. But because we don't want to classify variants based on just a single piece of data, even if it's functional, we downweight it. We downweight it to a +4 and force ourselves to find additional evidence that would combine with the +4 to push it to the likely pathogenic at +6. So just a degree of caution, a degree of conservatism there to make sure we don't make mistakes. But the take-home message here, if you look at this complicated Sankey plot, we show the different types of mutations vertically, or variants vertically. We break it down into all the little numbers and what we found in the functional assay, and then we combine it with all the other available phenotypic data and genetic data. And what do we end up as classification? Well, out of many thousands of variants, almost 7,000 variants, we end up with only 611 that remain as VUS. And they come from all categories of variant, all type of variant, but only 611 remain unclassified. Over 90% clearly end up in a category of classification, whether it's mainly likely benigns and then some likely pathogenics and pathogenics. And you'll see below that that's almost 800 variants that end up as likely pathogenic and pathogenic. I will comment, though, that some of these are, of course, truncating mutations, nonsense mutations that contribute to that group. So that's not all missenses. There are about 400 or 500 missenses in that category. But when you consider, if you go into ClinVar right now and you look for how many missense variants are considered pathogenic in BRCA2 or in this region of BRCA2, it's really not very many. So to be able to add 500 to that is really quite helpful in the clinical setting. But there are limitations to this. As I said, there's about 5% error. And it turned out that when we published our paper in Nature, another group from the National Cancer Institute at the NIH, Shaim Sharan is the lead PI there, they had done a very similar sort of experiment using embryonic stem cells. So these are humanized mouse embryonic stem cells. Again, using very similar technology, they're inserting the point mutations into the germline, and then looking at functional effects, looking at the same viability that we looked at, and really calibrating it in very similar ways. Their sensitivity and specificity relative to controls was a little bit lower. So 89% and 93, whereas ours up above here is 93 and 95. So there is a difference between the two assays. But it's an opportunity here where you have two very large experiments of the same set of variants to really maybe combine that data or use it in some way to improve the final classifications. So we decided to actually try to do that, right? Could we take these two models, combine them in some way? One way to do that is what we call the concordance model, where you literally just take the data and slap it together and look for consistency, right? Do the two assays both show me that that variant looks non-functional? Okay, then I would believe that. But if one is non-functional and one is functional, then I call that discordant and I throw it away. And that's why you see here we say we could only evaluate 80% of the variants because there were 20% that were showing us discordance between these two functional assays, which is quite a lot. The model we ended up focusing on is the VARCOL model. Now, the VARCOL is just the name of the statistical model that we use to evaluate our data. And it's developed by Ed Iverson at Duke University. And what it really focuses on is batch correction. It also accounts for variation in the replicates. So if you have let's say two experiments, two experiments on the left and one on the right, and they're a little bit separated, it downweights the one that's separated away from the other two. So it's looking for consistency in the data, and it models the data a little bit as a result. So there's a number of advantages to VARCOL over what we call the standard GMM model, which is basically a two-component mixture model, to develop those distributions and to classify the variance. So we took the VARCOL model and we integrated the data. So how did we do that? So essentially, because the experiments are so similar, we were able to take their raw data and recalibrate it onto our system so that it became essentially two additional data elements in addition to our triplicate data. They only had duplicate data, we had triplicate, but we could organize the data such that and normalize the data such that we now had 5 data points. And then we reran the VARCOL model. And so that was it's quite an interesting experiment, but it allows us to evaluate essentially all the variants with the advantage of having the 2 different experimental approaches embedded within that analysis. The other approaches down below are just sort of secondary approaches to the same things. So what did we find? Well, the graph on the left shows a comparison with, again, ClinVar standards, known pathogenic, known benign missenses. And again, you can see on the left that the integrated VARCOL model performs really quite considerably better than the concordance model. Now, you might look at the percentages and say, well, they're not really different, right? 95 versus 99, okay, it's only a couple of percent. But that's hundreds of variants. And that makes a big difference to the patients when you're potentially getting things wrong versus getting things right by doing a more thorough investigation. Some of the other secondary models are quite a bit lower in terms of their sensitivity. And on the right-hand side, we use a totally different set of standards, which are DNA repair assay variants. So we have about 500 variants that we've individually run in a DNA repair assay. We can use those in an alternative set. And again, you can see that the performance of the integrated VARCOL model is really quite superior compared to the others. So that was interesting to see that we can combine the data. We can come up with perhaps a slightly better result. And so what happens when we put that back into an ACMG model? You end up really reclassifying and changing a number of the variants. And the bottom line in text here is probably what's most important. 175 additional variants were classified as pathogenic, likely pathogenic, compared to that concordance model where you just throw the 2 data elements together and look at discordance. So really there's a benefit to this integrated model, and I think it really advances our cause here in terms of classifying even more variants and more accurately classifying the variants. Still a degree of error there, still a couple of percent where there's probably error in there somewhere. And again, that's why it's necessary to downweight that functional assay to a +4 instead of a +6 or a +8, okay? We're still playing that game, trying to balance things where we're not overemphasizing the function, even though we now are much more confident in it by having 2 assays combined together. So to prove that all these variants that are coming out and look like they're classified and look like they're quite attractive, we wanted to just run case control association studies to verify that the variants we're calling pathogenic in this setting actually have high risks of breast and ovarian cancer. And that's what you're looking at here is on the left-hand side, we're essentially looking at breast cancer, and we have data from a large testing company. And we can run, and in addition, there's 2 studies. The second one is a population-based study. So we can look at 2 measures, one from a high-risk testing population, the other from a population-based series. And you can see that the variants, the missense variants that we call P strong, so likely pathogenic, have that high odds ratio for breast cancer in this setting, very consistent across the 2 studies. And you can break it down as you go. If you look at the ones that were ACMG classified, again, very similar odds ratios. If you look at known pathogenic standards, very similar numbers again. So everything, and then of course, truncating mutations as a control here, and everything lines up very nicely. So it does tell us that the results from this functional assay really do seem to be reflecting that increased risk of breast cancer. And when you look at ovarian cancer on the right-hand side, you see exactly the same thing. Those points are all very much in a line. So really it looks like the functional assay is giving us clinically relevant variants. Sorry, didn't advance. Try that again. There we go. So that's sort of one of the take-home messages from my presentation here today, is that we've done these large functional studies. We've combined them together and integrated them together to improve the classifications. And then we can confirm that, in fact, they increase risk of breast and ovarian cancer. So for the most part, I won't say perfect, but for the most part, a lot of these variants could be considered to be sort of nailed down, at least at the functional level, and then ACMG models. But there was another question that remained. What about these hypomorphic and intermediate variants? So if you remember the original distribution, I mentioned that there was this little group in the middle. A lot of people would look at that and say, well, that's just error in the system. That's maybe your 2 to 5% error that we can't account for. That's possibly true, but we do know that it's a biological system and it should have 0 to 100% activity, different variants all the way across the spectrum. And so these may be the ones in the middle at around 30% to 50% activity. There have been a number of hypomorphic or intermediate variants reported previously. Most of these are involved in RNA splicing, so they have a partial effect on splicing. They don't completely knock it out. So you get this partial effect on splicing, partial risk. And so they're coming in at around 25 to 30% lifetime risk instead of the 69% that I mentioned before. But there's only 3 non-splicing missense variants that have really been thought to be hypomorphic in this setting. So we wanted to look further to see if there were more, and we had this large data set available to us. So we had also, as I said, we'd run about 500 variants through a DNA repair assay. This is a homologous recombination DNA repair assay. And what you're looking at vertically here is the amount of DNA repair, and across the bottom are all the different variants. And the ones in red at the bottom right-hand corner are known pathogenics, missenses. The ones in green are known benign missenses. And then we have this group in the middle that are sort of in no man's land here. And you can see this comment about the biological system where you have a spectrum all the way across the different ranges of activity. So we looked at these 70 variants and said, well, these are potentially hypomorphs. Can we do a little bit more to verify that? So we actually looked at them in the large MAVE assay that I just talked about, the large BRC2 functional assay. We looked in there, and they did look to be in the middle. And then we actually ran a controlled experiment where at each position of each of those 70 hypomorphs, we made the hypomorph, we made a stop codon, and then we made a benign at the same amino acid, so that we were normalizing for position effect within the gene. And then we reran that CRISPR study by integrating all of those variants. And this is what the result looked like. Again, you're looking at the functional score. So on the left, in this somewhat purple color, are your nonsense or protein truncating mutations. On the right, in the light blue color, are your synonymous variants that don't change amino acid. And right in the middle are these 70 hypomorphs. So here's a completely separate biological system. Originally, we observed them in a homologous recombination assay. Now we're looking at them in a viability assay in an endogenous knock-in system, and we get exactly the same result. So it was essentially confirming what we found. And then we also looked at RAD51 foci in the setting of each of these 70 variants. And you can see on the right-hand side, I just show a couple of examples, but we see a nice distribution. And we do, in fact, verify the hypomorphic activity in that setting. And then to just verify that, in fact, they might be causing intermediate levels of risk, we combine them all together in another large case control association study. 2 different large testing companies now providing their data to us. The top one matched against nomad controls. The bottom one actually has an internal set of controls within the company's data set, so perhaps a little bit better normalized here. But again, when you look over on the right-hand side here, if you look at the hypomorphs in general, you get 1.5. But if you look at that stronger half, so if you took that middle group and you took the stronger bit, you get an odds ratio of 2.4. Now, truncating mutations and missense standards, known pathogenic missenses in this setting are giving us 7 to 9, but these are giving us 2.4. Okay, so then we go down to myriad genetics and we look at that strong group again. What do we find? We get 2.1. And in their hands, the truncating mutations are 5. So we're going from very high risk for the protein truncating all the way down to slightly more than 2, odds ratio of 2, in the setting of this group of hypomorphs. We can't look at them individually because they're so rare in these populations, even in these huge testing data sets. They're too rare to look at one at a time. So we had to pool them together. But it is sort of a proof of principle that these things exist. So then we tried to model the cumulative lifetime risk here. And it's maybe hard to read this on the screen a little bit, but let's say on the left, breast cancer. On the bottom, in the blue line, you're essentially looking at the low risk of a benign variant. This is the population-based levels of risk. And up on top here in the sort of red color, at least to my eyes, you're looking at what looks like a protein truncating mutation coming in this setting at around 60% lifetime risk. But here's your hypomorphs in the middle in this green color. And they're coming in right at around 20 to 25% lifetime risk in this model. Over in the ovarian cancer setting, they come in around 4%. Now, in the United States, our thresholds for enhanced MRI screening in the screening setting for mutation carriers is about 20% for breast and about 5% for ovarian. So here we're at 24% and we're at 4%. So in this setting, the hypomorphs have much lower levels of risk, only about 25% lifetime risk in breast, but they still cross that magical threshold of 20% and would still qualify for enhanced screening with MRI. Sorry, just trying to get it to advance. So in summary, what have I told you today? Well, that we have functional assays now that have allowed us to look at this key DNA binding domain and that we were able to classify 90% of the variants in there. But when we combine two large CRISPR studies together, the quality of the results improved substantially. These strong variants that I'm talking about, these pathogenic strongs, are associated with very high risks of breast and ovarian cancer, just like protein truncating mutations. So they're probably clinically relevant. Validation using the VCEP guidelines is now needed. So just because I say that I ran it through an ACMG model for classification, that's a first pass, but I'd like to see the bodies such as CanVig, such as Enigma, such as ClinGen, taking our data and rerunning it through their systems to verify what we found. I think it will verify for the most part, but it still should be done formally. I think further studies on all variants in BRCA2 are needed. There's much more the rest of the gene. There will be rare, very rare pathogenic mutations hidden in there, mostly affecting splicing. We really don't have a lot of evidence of other regions of the gene that are important for function and that drive risk. But there will be a few hidden in there, and it's worthwhile doing that. And of course, further replicate experiments would help. I showed you 2 large functional assays combined together. If we had 3 or 4 or 5 of those very large screens all combined together, the data would probably improve in quality. We would make less mistakes. There are a few mistakes in there, but that costs money and it costs time. So we'll see what people decide to do in that setting. And then also we have these hypomorphic variants that I told you about. I think it's fairly clear from the data I showed you that a series of functional assays all show that they're intermediate in function. They do appear to be intermediate in risk for both breast and ovarian cancer. And they are now a complicating factor because they do suggest that there's a bunch of these variants out there that we will need to modify the risk assessment and management guidelines because, again, would you do a prophylactic surgery on someone who has a risk of breast cancer equivalent to maybe a CHEK2 or an ATM variant? In the United States, that would be contraindicated. We would not do that. We would do more screening. So it raises a number of questions and it raises the need for guidelines for a number of these variants. And we've done additional studies now. The 70 were just a proof of principle. We've done additional studies. We think there's as many as 300 in BRCA2, and there may be similar numbers in BRCA1. So this is actually going to be an area that will need some careful thought and some careful experimentation. And I will just thank the people who did the work. Most of the work that was shown here was done by one graduate student, one PhD student in my lab, Waji Huang, likes to be called Gilbert. We had a senior scientist helping them, and the rest of the people here are bioinformatics and statistics support. And then our collaborators, including Shaim Sharan at the NCI, who gave us the data from his parallel experiment, Ed Iverson, who developed the VARCOL model, and other collaborators. So I will stop there. Thank you.
Thank you, Fergus, very much indeed. So that was a very insightful and, as you say, a huge amount of work there. So thank you for sharing that in such a clear manner. Thomas, can I now come to you, please, and hand over to hear about the review of the learning that we had from Run 14, which was delivered earlier this year.
Thanks. Yeah, thanks for a very, very interesting talk, Fergus. I mean, that's really amazing data, but I will continue with the learnings from Run 14. And I will start with to give a brief overview of the results from run 14. So next slide, please. So 359 participants completed all variants, and of these, 181 participants correctly assigned the pathogenicity of all 6 variants. So that's shown in the figure to the left, and it's slightly more than 50%. And another 115 participants had 5 variants correctly assigned. And to the right, you can see the individual variants, and you can see that all of them are correctly assigned as either class 1 and 2, class 3, or class 4 and 5 by more than 85%. And I think overall there is a significant progress compared to previous runs. Okay, next slide, please. First, I want to acknowledge the work by George Burgell and Andrew Morani, and all the following learning points from run 14 are based on their summary. So next slide, just to show the variants included in run 14. There was 2 ATM variants, 1 missense variant and 1 frameshift variant. Then there were 3 BRCA1 variants, 2 missense and 1 frameshift, and finally also 1 frameshift BRCA2 variant. So next slide. First, I will go through some learnings regarding classification of ATM variants. And the first criteria I will touch upon is PM2 absence in control populations. And here is one important note that the ClinGen version 1.3 guidelines released in March 24, and the publication by the Hereditary Breast, Ovarian and Pancreatic Cancer VSEP published in September 24 is not identical. So in the publication, PM2 supporting can be applied if absent in GNOMAD version 2.11 or present below or equal to 0.001% in all subpopulations. While in ClinGen version 1.3, the frequency should be below or equal to 0.001% if n equals 1 in a single population. So in this case, there's no population data set specified. And in the next slide, you can see that this is also in the CANVIC guidelines, they recommend that you use GNOMAD version 2.11, the 2.11 data set. And this difference actually means that one of the variants, ATM variants, you actually could use PM2 supporting, but using the other set of guidelines, you could not. So it's really important to be aware that the guidelines that you are using. Okay, next slide, please. The next, I mean, this is actually just a point to follow, because there's just recently come some new guidelines, version 1.4, and regarding PM2, now it's clarified that you should use GNOMAD version 4 actually. So this is just for future schemes. Okay, next slide, please. Another learning from run 14 regarding ATM is the use of the benign evidence BP1. And BP1 can be used for missense variants in a gene for which primarily truncating variants are known to cause disease. And in this run, it has been used by some participants, but as you can see in the guidelines, it's not applicable for ATM variants. And that is stated in both the ClinGen and the CANVIC guidelines. So that is just for future use. You cannot use it for ATM. Next slide, please. Another criterion I will briefly touch upon is PM5, as a few participants did not use this criterion. And PM5 originally deals with novel missense changes at an amino acid residue where different missions change determined to be pathogenic has been seen before. But in some gene-specific guidelines, PM5 has been re-proposed to give extra weight to protein termination codon variants and PTC variants in an exon or a region where a different proven pathogenic PTC variant has been seen before. And for ATM, any frame shift or truncating variant with a PTC upstream of codon 3047, if you see that, then this criterion can be used as supporting. So that's just to know for future schemes also. Next slide, please. The final criterion I will mention regarding ATM is the PM3 criterion used for recessive disorders detected in trans with a pathogenic variant. And here different points can be assigned depending on the phenotype and whether the variant identified is confirmed in trans. The phase is unknown. The second variant is unidentified or VUS, or whether the variant is homozygous, as you can see in the picture to the left. And the data can then be combined for serine carriers, if possible, using the point system seen in the figure to the right. And it is mentioned for ATM variants that regarding phenotype, is actually stated in the clinician guidelines that publications that claim a clinical diagnosis of AT are taken as face value and granted a confident diagnosis, meaning that scores for phenotypes, phenotype confidence can be used, and that is shown in figure to the left in the upper part. So you can use that score and not the score just below consistent. And that actually means that if you have patients in the literature, you can use them and get evidence for PM3. The next slide, please. I will now continue with learnings regarding classifications of BRCA1 and BRCA2 variants. And the first criteria I will touch upon is the use of the benign evidence BP1 concerning missense variants in a gene for which primarily truncating variants are known to cause disease. And in contrast to ATM missense variants, BP1 can be used for BRCA1 and BRCA2 missense variants outside a clinical important functional domain with no predicted splicing effect. But it's at different strength when you compare the Enigma Clinician versus the Canwig. You can use BP strong when you use Enigma, but you can only use BP supporting if you use the Canwig guidelines. For Enigma, the guidelines also, this score also goes for silent and in-frame insertion, deletions, and Dillon's variants with no predicted splicing, and that is actually not mentioned in the Canwig guideline. And just to mention another homolog recombination repair gene, PALB2, you can use BP1 supporting for all missense variants, since very few pathogenic missense variants have been identified in this gene. Next slide, please. Another criterion where there is a difference between the Enigma Clinician and the Canwig guidelines is BS1, and that's the criteria where you look at the allele frequency and it is greater than expected for an disorder. And the Enigma Clinician recommendation is that you use the NOMAD version 2.1 or 3.1 datasets, the non-cancer cohorts, while Canwig uses the NOMAD 4.1 dataset focusing on females. And moreover, Enigma Clinician employs two levels of strength for the use of BS1, and that is BS1 strong, but also supporting, while Canwig only employs BS1 strong. Okay, next slide, please. Also regarding the criterion PM2, that is absent from controls in population datasets, there are differences between the Enigma Clinician and the CANVIC guidelines. Again, different data sets are used. For Enigma, it's the NOMAD 2.1 and 3.1 non-cancer. And for CANVIC, it's the NOMAD 4.1 data set. And in the absence of any carriers in the NOMAD 4.1 data sets, you can, using the CANVIC guidelines, use PM2 at a moderate level, whereas only supporting level is used by Enigma and Clinician. Moreover, Enigma Clinician states that this criterion should not be applied to insertions or deletion or del-in variants, and also that the read depth should be higher or equal to 25. And these restrictions are not mentioned in the CANVIC guidelines. So here's also some differences that you should be aware of. Okay, next slide, please. One of the final differences that are seen in this run is the difference between Enigma Clinician and CANVIC when you use the BS3 criteria for the benign for functional studies. And in Enigma, you have a table in table 9 where you can go in and look for specific variants and see the evidence score. And when you go and look for one of the variants in this one, you can see that you can use BS3 as strong. But when you use the CANVIC review functional assays and scores, you can only for the same variant use BS3 moderate. So again, there is a difference here, and that actually leads to the final slide. Next slide. Which is actually the key message is that that different variant classification guidelines may lead to different conclusions, and variant classification guidelines is still evolving, and therefore it's important to indicate on reports which guidelines and which versions that were followed, and also be aware because the published guidelines and the most recent guidelines may not be identical, and you can find the most recent guidelines in the Clinician criteria specification registry or at CANVIC shown below. And I think that was what I had, and more comments to run 14 can be found in the summary report. Thank you for your attention.
Thank you so much, Thomas and Fergus, for a really, really good webinar. I think you've had two different perspectives there from both the sort of the background, the science behind how we investigate the classification of these variants, and also the impact of the work that GenQA and EMQN have been doing on the practical components of laboratories investigating the variant classification. So just leaves me to sort of draw things to a close before we do the question and answer session. So if you haven't yet enrolled for the next run 15 of the HRR and BRCA1 and BRCA2 gene variants classification EQA, please do head over to the website there and log in or register, as Sandi explained in the start of the webinar. The assessment process itself will be taking place from the 1st to the 31st of October. So you've got 2 weeks or so to get yourselves registered and head over there to get signed up for the EQA. Any questions or information you're lacking or require on it, then please do contact both the EMQN and GenQA teams. Next slide, please. Okay, so this is probably the most, after the learning you've got from the two presentations there, this is probably the most important part of the webinar and the bit where we really need your participation. It's obviously your opportunity to raise questions for our speakers. So myself and Sandi are going to manage the questions, and we've got quite a lot coming in at the moment, which is fantastic. So I'm going to kick off with a question for Fergus, if that's okay. Fergus, you presented some fantastic science around investigating the functional effects of these variants in BRCA2. what came across to me, apart from the huge amount of work that you've done, is also how complex that scientific experiments were. How realistic is it to extend that study to other variants in the entire BRCA2 gene, and by extension to BRCA1 and other HRR gene variants?
Yeah. So we're not doing the rest of BRCA2. It's a huge experiment, costs a lot of money. But I believe that David Adams' group at the Sanger Institute, just outside Cambridge, is actually planning to do that. So I think it will get done by a really high quality group. So look forward to seeing that myself, in fact. Now, extrapolation to the other genes. It's all in the works, actually. BRCA1 was reported actually first about 4 years ago in a Nature paper from Leah Storita's lab. I haven't seen any additional MAVE experiments from other groups. It would be nice to see additional CRISPR studies done, just for the comparison, like I was showing earlier. If you had 2 or 3 different groups did it, it'd be nice to put the data together. PALB2 has been done, and there's a paper in review right now. As was commented, there's a relatively small number of pathogenic missense variants in that gene. It looks like slightly more than 100 across quite a large gene, so small numbers there. CHEK2 has been done. A couple of different studies done in yeast actually have been published, and there's an effort ongoing right now, actually within the Enigma group, to combine all that functional data together to get an overall score and combine it then into the ACMG models. So that's ongoing. ATM, there was a paper recently reported from a group in South Korea, a very large in Cell, where they described the whole gene, evaluating the entire gene, which is a huge undertaking. There's, I think, 67 exons in that gene. So massive amount of work. So that's they've done that and thrown it out there. We've done actually some of that data, and we find, okay, there's only about 80% correlation between our data and their data. So maybe, you know, there's a need for further experimentation in that gene, but there's some data out there currently. And then RAD51C and D, David Adams' group, again in the Sanger Institute, reported on RAD51C functional studies early at the very beginning of the year, I believe. And so there's efforts ongoing now to integrate that into an ACMG model on a large scale, you know, all the data basically, and see what that looks like. And RAD51D, there's some studies ongoing there. So I think, to be honest, of the ones you think of as HRR genes, I think they're all essentially done in terms of these large functional screens. And now it's our job to actually integrate all of that into the classification models. And as was pointed out, you know, there is error in these experiments, right? So we do have to be a little bit cautious with this stuff. And it's always nice to have additional functional assays, even if it's just single variant functional assays where they're being spot checked. I think that would be quite valuable for most of these.
So, I mean, it's fantastic to know that most of the genes have already got at least something being worked on them. I've got a question from another one of the questions here. So, you may have multiple partly independent functions of BRCA2 contributing to cancer development. Therefore, different functional assays may be discordant but still meaningful. How can this be taken into account when integrating data from multiple independent functional assays?
Yeah, that's a great question. It's always in everybody's mind when we do these things. For us, we're fairly confident that we are measuring the correct functional assay because of that odds ratio calculation that I showed you, right? Where we show that with the variants coming out of our functional assay, we get very similar odds ratio to protein truncating variants, et cetera. So it does seem to suggest that as a group, the variants that we select as being pathogenic are the high risk, increased risk. Now, does that mean we're not missing a few that might have been pathogenic in a different assay? That's certainly possible. There's just, I think we know enough about, let's say, BRCA1 and 2 to begin. We know enough about those proteins to know if we're missing a function or not. I think we've got it covered. The one thing where we might have a difference is not so much in the risk, but in response to therapy. It's possible that drug response could be different to risk. Just these functional assays may read out differently in that regard. And so it concerns me a little bit when some people are using drug response as the functional assay to predict risk. because now you're lumping two things together, right? You're looking at viability and risk, and you've got a drug response, and they could be different. So it causes a bit of a mixed message there. Sorry. So BRCA1 and 2, I think we're confident. Genes like PALB2, CHEK2, ATM, where they are known to have multiple functions, these could be a little bit more complicated, I will admit that. And so there is more work to be done to sort that out. I mean, the only way to get the answer is to do the experiments, right?
Yeah.
And we'll just have to see how it goes.
And also then obviously the last bit on your data was really the role of hypermorphic variants as well. And clearly that's a sort of emerging area as well in terms of impact.
Right.
And your studies seem to be at least addressing that for BRCA2. Do you think people are finding similar findings for the other genes, the other HRR genes at all?
We're not seeing a lot, to be honest. The PALB2 data, we're not seeing a lot of hypomorphs. Again, there's so few pathogenics anyway that there's just not a lot there. The CHEK2 and ATM, I'm not sure it matters too much because your risk of breast cancer is so low to begin with. Like if you're only at an odds ratio of 2.5, lifetime risk of 25, then what's a hypomorph in that setting, right? Is it an odds ratio of 2? It's probably still clinically actionable. Is it an odds ratio of 1.8? If you had an odds ratio of 1.8, would you not deal with it? I mean, again, I think a hypomorph in the setting of those genes will likely be treated the same as a full pathogenic in those genes. In the setting of breast cancer, which of course it's a moderate risk, but now you take some of those genes like ATM and you take it into pancreatic cancer, now you've got a high risk gene, right? So it may actually behave quite differently there, but there's just not enough data in the pancreatic field just yet to determine how these things might be behaving. Still plenty of work to do, that's for sure.
Well, that's reassuring and good to hear. Sandi, over to you.
Lovely, thank you. Thomas, I've got some questions for you. As always, we've got some questions about particular guidelines and different criteria. So to kick us off, the CanVig UK guidelines for CHEK2 refers to the guideline for ATM gene. So would the presented information, so what you presented, apply also to CHEK2?
Yeah. I mean, I do not use the CanVig myself. I mean, I use the Enigma Clinton guidelines, but as I read them, I mean, I would think they as stated in the ATM that should also go for CHEK2. So I would read it in that sense. So when they read as written in the ATM guideline, they should read it like that just for CHEK2.
Because that is what is being published related to that gene. So we always try and address this on our webinars, is about the different guidelines that are available and to be applied and when. So somebody's asked, why is there such a difference in the Enigma versus the CanVig classifications? So I guess you can't answer this on behalf of the two sets of authors, but from your point of view.
but I agree. I think it's a problem, right? Because I mean, it makes it more complicated, and also if you look at the same variant and you get different classifications using different guidelines, I mean that's silly. And I know that different groups are working and doing their own studies for specific genes and thereby also use the rules a little differently, but my hope is that we are waiting for the general guidelines to be updated, and that would help. So they will be more equal. I mean, in the end, there should, in my opinion, only be one guideline for each gene, right? But
it would make life an awful lot easier, wouldn't it, for standardization and even just people's competency and confidence to be able to know that they're doing the right thing. One question, bit more general. So about the CanVig guidelines, is there a defined list of cancer susceptibility genes that can be classified using the guidance, or is it basically all genes associated with cancer? It's quite a tricky one to answer that.
I mean, for the CanVig, I mean, there is a lot of gene-specific guidelines for cancer genes, right? So of course you should use them when possible. But if there are not gene-specific guidelines, I guess for CanVig you could use the general guidelines also, if that's the question.
Yeah. Yeah, I think it was also, is there a go-to list of what a cancer susceptibility gene is? I think you would just have to do a review research,
Yeah,
PubMed, and just find out if you had a query about a particular gene you were looking into specifically. Would you agree?
yeah, because I mean, there's not all the guidelines are not made yet. So I mean, there's
Exactly.
several cancer genes where you don't have gene-specific guidelines, and we are of course waiting for those. But I mean, in reviews you can find the cancer susceptibility genes that you need to and also in your gene panels. I mean, for which genes you have to look for. So
Yeah, and I
yeah,
guess that's where professional sort of experience and comes in itself, isn't it? Your judgment, your professional
yeah,
judgment. Yeah.
yeah.
Right. Simon, back to you for a few more questions.
Thanks, Sandi. Fergus, just one little follow up on your presentation. So I think one of the delegates has asked whether the published data is available. Could you provide a link to the published data, please, so we can and we'll share that more broadly with the participants.
Yep, I can do that. And then there's also a question about what are the 3 missenses that are
That's
consensus.
what I was going to come on to next.
Yeah. There was a paper published, but I can give the link to that too. I can provide those.
Great, yeah.
Fabulous,
Thank you.
Yeah. Okay, so that's just answering there was a question there about the 3 BRCA2 missense variants that consider the consensus hypermorph. So we'll provide a paper for that. So we can provide the background information for the delegates. Sandi, over to you.
Yeah, right. I'm going to come back at you then, Simon. So this is an EQA question, really. So what recommendations does EMQN and GenQA give regarding reporting exons deletions in the BRCA1, BRCA2 genes in medical reports? So I think we see this in lots of our EQAs about how you use nomenclature, standardized nomenclature, to report breakpoints, I guess, of deletions and duplications, although you've got tandem duplications to factor in there, using NGS, MLPA, main transcript. Do you apply HGVS? What's the focus for the EQA providers?
So our focus has traditionally been around clinically relevant nomenclature guidelines. So we would utilize the guidance from established bodies like HGVS, etc., for ensuring that the interpretation is carried out correctly. But more importantly, on a general perspective, we also look at how that information is conveyed on the report as well. So we'd also use guidance from, for example, published best practice from the European Society of Human Genetics, for example, on how that information is conveyed on the clinical report. And the combination of those 2 factors, the nomenclature guidelines and how to actually utilize to report these variants, and the reporting guidelines themselves, gives a pretty good structure for laboratories about how to convey that information in a clinical report. Would you add anything else to that?
I would say probably when it comes to describing deletion breakpoints, the current European Society of Human Genetics guidelines for reporting is very much make it simple and less likely to be misinterpreted. So if you can actually use exon numbering, where you can be assured that that coding sequence has been deleted, then we would rather see a written description. We do see, as you know yourself, Simon, many errors popping up in a range of EQAs, whereby people try to describe using where the MLPA probes end or from their NGS data, and not accurately describe the actual deletion, or don't put caveats in to say that it's a minimum deletion because you don't know what's happening in the introns, and also using exon numbering that's not linked to actually a reference sequence that's current, it's discrepant. So I think be on the safe side for patient safety to make a clear written description of what you're actually seeing and make sure that it's accurate.
I would agree entirely with that. I think the clear thing is about clarity and making sure that it's as simple as possible. But I also, in defense of laboratories as well, I think we need to acknowledge that the field around this, particularly around reference sequences, it has been blurred by the changes to the implementation of different reference sequence standards. So the latest being the main transcripts, having gone from RefSec, where we spent probably 10 years of trying to ensure we had standardization with respect to laboratory reporting on it. So I think we, as EQA providers, also we have to have a level of pragmatism in our assessment of the laboratory's practices to deal with the fact that there's so much variability around it, and the complexities as well of obviously now, you know, understanding the genome in more detail and maybe able to interpret different specific variants as a consequence. So pragmatism is also part of our work, isn't it, as well, to make sure that we are reasonable about how we manage the implementation from the laboratory side of things.
Sure, yeah. And education, because that's what EQA is about,
yeah,
isn't it?
Yeah,
So
absolutely.
we're all learning from each other.
Yeah,
Yeah, exactly. Right, thank you for that. Thomas, I've got another couple of, sorry, I know these criteria, they always come up and bite us, don't they? So please clarify the use of nomads. So for PM2, this is. So is ClinGen Enigma meaning the use of nomad version 2.11 and version 4, or is it nomad version 2.11 or version 4? So do you use them together or as separate entities?
I mean, you don't, as you use them, if I understand the question correctly, you use either one. I mean, for the BRCA genes, it is the non-cancer cohort that they're interested in, and I don't think that you have that for the version 4 cohort. So it's either 2.1 or 3.1 non-cancer. So that's the recommendation there. But in Canvig, the recommendation is different. And they look at the 4.1 and then only females, right? So at least for BRCA1 and BRCA2.
Yeah, I see Fergus nodding as well. Fergus, is there anything you want to add to that?
Yeah, I think, you know, it's not on the website yet, but certainly the Enigma BRCA1/2 VSEP, I think is moving to V4.1 as well.
Yeah.
So it'll be more in line with Canvig going forward.
Yeah, that's good to know. Do you want to, Thomas, just maybe give a really quick summary of what's different between the two data sets?
It's I mean the 4.1, it's bigger and it has all the UK biobank data there. So I think that's the main difference between the two.
Sure.
And as I understand it, some of the phenotypes are not quite clear in the UK biobank, so some people will not use it, but that's I'm not sure about that. But at least it's much bigger, and it would be nice if all, of course, use that cohort. Yeah. And then, of course, when regarding the PM2, I know that the new guidelines are discussing whether you should give it any point at all, right? So maybe you will not use that population data, at least for in the pathogenic view, but only in the as benign evidence in forthcoming guidelines.
Thank you. And then we've got one final question that's come up is concerning the PS4 parameter. When using the Fisher exact test, does it matter which website you use for calculation? And the individuals put two in here. One declares Turnbull's lab and also a stats info page. Any thoughts on that? No.
Really, maybe that should be something for the report, but
yes,
I'm not sure.
Maybe we need to go and ponder that and maybe put some figures in and see if there's a difference when you, or different criteria used. Yeah, I think we'll take that one back as a to-do, Simon.
I think so, Sandy. Let's definitely do that. Okie dokie. I don't think we've got any more questions, and I don't wish to put any more to our speakers at short notice, or lack of notice, should I say. So I think, shall we draw it to a close, Sandy? Okay?
I think so. I think it's been incredibly educational and enlightening. I've learned so much on this webinar, so a huge thank you to Thomas and Fergus very much indeed for all your preparation work and today as well. Yeah, just to echo my thanks as well to you, but also to the participants for dialing in. Thank you for your time. You're giving up your valuable time during the day to listen to our webinar, and the fact you've done so is fantastic for us. Please do try to complete the survey. We've provided a QR code on the screen here. It takes less than 2 minutes, does help us in terms of being able to make sure we provide relevant future webinars for you. A big thank you also to the organising team. Thanks for your hard work to getting this webinar up and running. And finally, just leaves me to thank my co-chair, Professor Deans, my colleague, and from myself, thank you again once more for joining us. And we'll look forward to speaking to you on our 4th and final webinar later on in the year. Thank you for your time. Thank you, everyone. Bye-bye.